Good morning, everyone from Fort Collins, Colorado. My name is Diana Wall, and I am the science chair for the Global Soil Biodiversity Initiative. We're really pleased to have a great webinar today. We're looking forward to it, but just a few housekeeping rules. First of all, we want you to know this is being recorded and it will be available on YouTube to everybody who registered. So tell your colleagues that it's available. Also, I just wanted you to know that you can put your questions and answers in the Q&A box, but don't be surprised if the panelists don't answer them. There's going to be a lively discussion, I'm sure. So I don't have any other announcements except to welcome you and to also welcome our moderator, Dr. Bruce Hungate. Bruce is from Northern Arizona University, and we're really pleased to have him moderating this morning. Bruce?
Welcome, everyone. Once again, please remember this is being recorded. That includes the chat. Recording will be available on YouTube. Let me introduce our four panelists. Good morning. I'm joining you from Flagstaff, Arizona, so it is definitely morning here. Dr. Kate Buckeridge conducts research at the Luxembourg Institute of Science and Technology. Her research focuses on the question, when a microbe dies in the soil, what happens to that microbial cell and all the molecules inside it? This brings up microbial necromass, which is a great word, which refers to the dead remains of microbial cells. Dr. Buckeridge's work has shown that microbial necromass is extremely important for soil organic matter. In her paper, which I love the title, Sticky Dead Microbes, Dr. Buckeridge shows how necromass becomes stuck to soil particles, an important way that soil organic matter stays in the soil. Dr. Buckeridge views soil organic matter through a microbial lens, an approach that has contributed to the mounting evidence that soil organic matter is a microbial graveyard. Thanks for joining us, Dr. Buckeridge. Dr. Gerrit Angst is based in the Czech Academy of Sciences, where he investigates the buildup, composition, and turnover of soil organic matter, and how its molecular composition relates to its persistence and stability. He's shown how the type of rock from which soils form, so-called parent material, determines soil organic matter accumulation, and how that organic matter is largely derived from microbes. His interests extend to the macroscopic, like plants in producing soil organic matter, and to earthworms in processing it. Dr. Angst has shown that when earthworms work their way through soils, the trail they leave behind, the casts, actually help preserve microbial necromass. Dr. Angst seeks all this knowledge as necessary to understand and manage soil carbon. Thank you for joining us, Dr. Angst. Dr. Cynthia Kallenbach leads her research team at McGill University. Dr. Kallenbach's work has provided strong, direct, and elegant evidence that soil organic matter is mostly microbial remains. You can see this is a theme this morning. Her work has contributed much to our understanding of how microbial physiology and mortality affect the buildup and loss of organic matter in soil, how diversity in soil microbes affect how plant litter decomposes, and whether carbon stays in soil or is respired away. Through her work, Dr. Kallenbach seeks to inform agricultural management that is both environmentally sustainable and productive. Thanks very much for joining us, Dr. Kallenbach. Dr. Colin Averill conducts research at ETH Zurich, focusing on the global ecology and biodiversity of microorganisms in soil and other organisms as well. Dr. Averill's work shows that the property of soil organic matter depend on how much bacteria occur in a given soil compared to fungi, and also depend on the particular type of fungus and its association with plants. Dr. Averill studies mycorrhiza, which is a beautiful word we can take apart to remember what it means, myco or fungus, and rhiza or root. So the word mycorrhiza actually means an association between a fungus and a root. We'll hear more about this. Another important aspect of Dr. Averill's work is that the effects of these fungus root associations on soil organic matter are apparent at very large scales, even global. So Dr. Averill's work really demonstrates that the global impacts of soil microbial biodiversity. Thank you for joining us, Dr. Averill. Thank you all the panelists for joining the discussion today and lending your world-class expertise to this important topic. Okay, a bit of introduction to our topic today. Soil holds all kinds of treasures for anyone taking the time to look. These treasures are documented beautifully in this Global Atlas of Soil Biodiversity, which if you have not yet checked it out, please do so. The link is here. Download it today. There's another report we're checking out, also from the Global Soil Biodiversity Initiative, about the state of knowledge of soil biodiversity, another excellent resource on the topic.
Dr. Hungate,
Yes.
I think there might be something on your microphone that's making it a little
Thank you for saying so. Is this better?
much better. Thank you.
Oh, wow. Okay, thank you. So biodiversity is all the variation in life, different species, different physiologies, different evolutionary histories, different biochemical pathways, all are part of biodiversity. Life in soil comes in all domains: bacteria, archaea, eukarya like fungi and protists, and soil biodiversity does not end there. Of course, plants of all kinds are part of it, as well as many different invertebrate animals, from the microscopic to the very large, insects and earthworms, and all kinds of arthropods, as well as nematodes and many, many other creatures. Soil biodiversity creates and consumes organic matter, the fuel and the product of soil organisms. What's clear now is that understanding how soil organic matter forms and how it is lost requires diving into all the details of soil biodiversity. Some of these details are emerging with new techniques showing which organisms occur where and what they're doing. For soil organic matter formation and loss, we can ask who is involved, where, and how. This is like an underground game of Clue, the board game where you try to sleuth out who murdered Mr. Body, who did it, where, and how. In Clue, it might be Mrs. Peacock in the billiard room with a knife. Whereas in soil, it may be bacteria in a spruce bog with reduced efficiency caused by warming, leading to soil organic matter loss. Soil biodiversity really is this dramatic. In Clue, it might be Professor Plum in the dining room with a wrench. In soil, it might be mycorrhizal fungi in a conifer forest, whose extracellular lignin-degrading enzymes cause soil carbon loss as CO2 rises. In Clue, it may have been Colonel Mustard in the ballroom with a revolver. And in soil, it may be cyanobacteria in Antarctica, exuding extracellular polymeric substances that cause soil carbon to accumulate with warming. Or it might be predators like protists and some bacteria that create microbial necromass by eating other bacteria. This could enhance or reduce the accumulation of soil organic matter, depending on how long the food chain. The list of possibilities goes on and on. So death is central to the game, Clue, right? Which turns out to be appropriate as we learn more about the role of dead microbial bodies, necromass, in forming soil organic matter. It's now clear that this is a very important part of the story. And as we drill down with omics and isotopes and models and many other soil sleuthing tools, we can investigate the dual roles of microbial diversity in both creating and consuming soil organic matter. So the trick is to find out who, where, and how. We care because stabilizing carbon in soil is important for many reasons. One is climate change. Carbon in soil organic matter used to be atmospheric carbon dioxide, the leading cause of climate change. So we want to keep soil carbon in the soil. It's also possible to promote processes that add carbon to the soil. Doing so will take CO2 out of the atmosphere, slowing climate change. The challenge is that if carbon in soil is not stable, it can return to the atmosphere, accelerating climate change. We also care because soil carbon is vital for what soils do, grow plants. Agriculture and forestry depend on soil organic matter. We're in a new phase of discovery about this important feature of natural systems, soil carbon stabilization, and how it relates to soil biodiversity. And we've got a panel of world experts ready to share their ideas and insights. So we're going to start off with Dr. Buckeridge. Dr. Buckeridge, the Zoom window is yours.
Thank you, Dr. Hungate. Okay, so is that okay? Everyone can see my screen?
Looks good.
I'm good, thank you. I'm going to talk about microbial necromass and soil carbon and the influence of soil biodiversity. So I guess Dr. Hungate started to talk about this, but I'll just go over this. What is necromass? It, as this scanning electron micrograph shows you, it's this is a scanning electron micrograph of bacteria on a sand grain. So you can see some intact cells, but you can also see a lot of the mush that's around it, that extracellular polymeric substances. So those are the proteins, DNA, and polysaccharides that formed the biofilm that those microbes lived in, or they might have been part of the cytosol, or they could have been the enzymes that were released during the processes when the microbes were alive. But once they die, you have intact cells, you have this extracellular polymeric substances, and you have cell wall fragments. And of course, that's really difficult to separate from the plant residues in the soil. And so we've just learned over the past 30 years or so how to detect this and how to recognize microbial necromass. And we do that with a couple of tools like molecular fingerprinting, where we look at specific carbon functional groups or molecules that are representative of microbes, or amino sugars, which are part of microbial cell walls, but not plant cell walls, and can identify bacteria or fungi. Or my favorite method, where we grow microbes on stable isotopes, which labels the biomass and the necromass after we kill them, and then we add them to soil, and we can trace them through the system then. So what does this have to do with soil carbon? So this is the exciting part. There's this new paradigm that's come out over the past 10 to 5 years or so, and it's really changed the way we look at soil organic matter. Previous soil organic matter research focused really on how microbial biomass broke down plant litter and root carbon inputs to produce greenhouse gases, CO2 or N2O, for instance, or to produce particulate organic matter, or POM. And then there's been a growing understanding that it also, some of the carbon was stabilized on mineral surfaces, and that carbon was more stable. But nonetheless, the focus was on plant chemistry, and more difficult to break down a recalcitrant carbon was believed to last longer. But now we have a greater understanding that plant carbon, when it forms microbial biomass, it also forms necromass. And that necromass actually makes up 30 to 80% of the soil organic matter, depending what ecosystem you're in. And a lot of that is mineral associated or mineral stabilized, making it more stable. And interestingly enough, it's the labile carbon, or the really easy to decompose carbon, that now produces more microbes, more microbial biomass. They grow faster with a higher carbon use efficiency. So more of that carbon stays in their biomass and less is released as CO2, meaning they grow faster, they produce more necromass and more soil organic carbon. So now the focus, instead of being on plant chemistry, is on microbial physiology. A little bit of both, but microbial physiology is the new focus. And the literature is currently, this is the thing that I think is exciting, is currently focused on production of necromass and the stabilization, or the net stabilization. We look at the carbon that's on the mineral surfaces. But it's missing, actually. There's a lot of focus on recycling, which is the microbes eating the necromass, or the destabilization, when microbes eat the necromass off the mineral surfaces. And both of those things happen, and both of them are important. The carbon that's on mineral surfaces is often assumed to be forever stable. It's just more stable. So those are some important things that are happening. What does that have to do with biodiversity, though? So basically, if you have a different microbial species or a different microbial community, you have different properties or traits, and these traits determine how much carbon is stabilized. So some of the traits that we've already talked about, growth rate, turnover, and carbon use efficiency, are obvious ones, and those are well studied in the literature. Some of the ones that we don't talk about often enough and probably need to understand better is the spatial or life history traits of microbes, whether they live in biofilms, rhizosphere, or in soil pores, because those could influence how close they are to a mineral surface, for instance, and how likely they are to be stabilized. And something that gets only a little bit of emphasis in the literature is the necrobiome, the microbes that are eating microbial necromass. So not all microbes eat necromass because it does have lower energy supply than, for instance, plant root exudates, higher than other substrates. And so there are microbes that eat necrobiome, but there are microbes that eat necromass. And that might be also important for destabilization too. And then of course, there's also the dead traits or dead properties of necromass, which are related to biodiversity, that could influence the bonding strength of the necromass to the mineral surfaces, and things like necromass C to N ratio, morphology, or even potentially specific compounds. And so that's some of the work that the work that Dr. Hungate referred to, my sticky dead microbes paper talks about that a little bit. So I'm just going to end by saying some of the research gaps that I think are most important, but because this is still a fairly new field, there's lots of research gaps, but the things I think are really missing right now are a focus on recycling and destabilization. And the related to that, the necrobiome, who's doing that recycling. Microbial necromass nitrogen, the C to N ratio in necromass, microbial necromass, is a lot lower than plant residues. And so it's got an influence on the stabilization, but it also has influence on the persistence of both carbon and nitrogen in ecosystems. And I think that's something that needs more attention. And finally, the thing that I think we always need more attention is we don't have enough global change crossed with agricultural land management experiments because they're difficult to put in place. But those are going to have a big impact on microbial necromass and therefore on soil carbon and some of the soil carbon sequestration mitigation efforts that we're hoping to enact in our agricultural land. So that's my summary, and I'm going to pass the talking stick back to Bruce.
Thanks so much, Dr. Buckeridge. The necrobiome, I love that. Next, we have Dr. Angst. Take it away, please.
Yes, thank you. So do you see my screen? Probably, yes. Okay, so jump right in. So thank you everybody for being here, and thank you for the organizers to invite me to talk about some of my research on earthworms and carbon sequestration in the soil. So first of all, as an introduction, I will tell you how I came to this research topic. So I performed my PhD at the Technical University in Munich in Germany, also on carbon sequestration. And here we did a lot of spectroscopic techniques. You can see an NMR spectrometer here, for example, a NanoSIMS device. We extracted biomarkers for amino sugars or for microbial-derived compounds, for example, or plant-derived compounds. So a lot of soil chemistry, but no soil fauna whatsoever. So then I got to know my wife, who's Czech, and she drew me to the Czech Republic, which is why I'm now working at this in this rather nice building, which is the Institute of Soil Biology. And here people almost exclusively focus on soil fauna. And I asked myself, okay, why haven't I heard much about soil fauna and carbon sequestration before? And I did some research on this topic, and I found that particularly earthworms, or research on earthworms is rather abundant, perhaps because they are virtually present in every ecosystem, like apart from ecosystems with extreme climates like deserts or Antarctica, and they are so-called ecosystem engineers. And while reading the literature, I summarized the effects of earthworms, and like the major effects can be shown in this graph. And here on y-axis, you have soil carbon contents low and high, and no earthworms and earthworms, so earthworms present and not. And on the one hand, earthworms create aggregates, so they increase stable aggregate structures in the soil, which stabilizes soil carbon, so which consequently should increase carbon contents in the soil. And on the other hand, earthworms add easily decomposable mucus to the soil, which stimulates the microbial community, and the microbial community decomposes soil organic matter, which results in the emission of CO2 from the soil and should thus decrease carbon contents in the soil. And the net effect of these two opposite processes are not very well known yet, probably because the two effects are dependent on environmental factors, on the earthworm species involved, and also on the study period. And surprisingly, for me at least, I hardly found any studies on the molecular level. This was surprising to me because when I did my PhD, we did a lot of molecular level studies to reveal processes on smaller scales that are potentially obscured on larger scales, like when you just measure carbon contents, for example. And I said, okay, then let's incubate earthworms with soil and soil without earthworms and perform molecular level analysis. And indeed, we found that earthworm reworked soils had a different chemistry or a different molecular composition of the organic matter than soils that had not been reworked by earthworms. So in earthworm affected soils, we had an increased microbial necromass in rather stable fractions of soil organic matter or stable soil compartments like macroaggregates and microaggregates. And we also had a decrease in plant-derived organic matter and the more labile fractions like particulate organic matter. And of course, this was just the lab experiment, which is why we are now extending our research to more realistic conditions in the field. And I'm currently cooperating with Nico Eisenhower and Martin Schädler from the Global Change Experimental Facility, which you can see here in this picture. And in this facility, we can simulate future climates via these roofs here and the walls. We have different land uses, and we're able to deploy different earthworm communities, so different earthworm species, different biomasses. And one of our hypotheses would be that earthworm communities with different ecological strategies, so with different earthworm species with different ecological strategies, would have higher influences on carbon storage and dynamics than individual species or species assemblages with just one ecological strategy. And in the end, we hope to be able to better parameterize biogeochemical models, so to somehow include the influence of earthworms into these models better. And of course, to manage soils better as a carbon sink, which would be rather the far aim of my research. And of course, this is just research on earthworms, and there's a lot more soil fauna which has hardly been investigated in the nexus of carbon sequestration, and particularly on the molecular level. So there's a lot more research to be done. Thank you.
Thank you very much, Dr. Angst. It's fascinating stuff. We next have Dr. Kallenbach. Take it away.
I want to start by talking about plants because we have this tremendous body of knowledge over the last century where we can see strong impacts of characteristics of plants and how they influence soil carbon storage and formation. So for example, we know that plant growth and net primary productivity can have a strong influence on soil carbon storage. And of course, plant tissue quality, things like carbon to nitrogen ratio of plant tissue, that this can impact the storage of soil carbon, and even the diversity of plants and how plants interact, all have impacts on how soil carbon is formed. And if we think now about the importance of microbial communities and their direct contributions to this more persistent, stable soil carbon pool that Dr. Buckeridge has talked about, we can start asking the same questions about the microbial community in the soil. So the same questions that we've been focused on for plants, we can apply those same questions below ground and think about how differences in microbial growth, for example, might influence this microbial feedstock or necromass that could go into this mineral-associated organic matter pool. So not all microbes grow at the same rate. Some grow fast, some grow slow, some grow more efficiently, where they might use more of the carbon they take up to build more biomass relative to, say, an organism that grows less efficiently. And of course, we can imagine how differences in community composition or biotic interactions can influence the growth and activity of microbial communities that could have downstream impacts on soil carbon. And even something as simple as microbial biomass chemistry or the quality or stoichiometry of biomass, so the carbon to nitrogen ratio of microbial biomass, might impact the fate of that necromass, as Dr. Buckeridge pointed out, into the soil carbon fraction. And so just as an example of this, my lab is starting to do some work in waterlogged soils in Quebec. And what we're finding is that the microbial biomass carbon to nitrogen ratio of the living microbial community varies quite a bit over the course of flooding. So we see this increase in the carbon to nitrogen ratio during flooding, and then a decrease once the flood retreats. And so one of the questions we're interested in is what does this mean for the fraction of carbon that's stored in the mineral associated organic matter pool. And interestingly, we're seeing that the mineral organic matter carbon is also responding to flooding, but in an opposite direction as the carbon and nitrogen ratio of the microbial biomass. And so what we're thinking about now is whether or not these two responses are interacting. So in other words, is one responding to the other? To think about how changes in biomass stoichiometry, both uptake, but also the deposition of that necromass might influence microbial inputs to mineral associated organic matter. Considering that if we think about microbial communities, the living microbial communities as an important component of building and storing soil carbon, we need to keep in mind that they have this whole range of traits or characteristics, and that a species can have very different properties that they've developed over the course of evolutionary selection, for example. And these traits or characteristics can also vary under changing environmental conditions. And so we might then ask, is there a specific profile or set of traits that might be most supportive or optimal for building soil carbon? And so one of the things that my lab is working on right now is whether or not differences in arbuscular mycorrhizal families, so these are obligate plant root symbionts, whether differences in arbuscular mycorrhizal families impact how soil carbon is stored. And so for arbuscular mycorrhizae, their traits are conserved at the family level, where we get variability in things like growth rate, hyphal turnover, spore size, where Gigasperaceae, the family of Gigasperaceae, for example, behaves more like a spruce or redwood tree, where it takes longer to sporulate. Its growth rate is relatively lower compared to Glomeraceae, which might be more similar to sort of a weedy species like dandelion. And so my master's student, Caitlin Horsch, is exploring this idea using 13 carbon isotopes and looking at whether Gigasperaceae or Glomeraceae, which have these distinct trait profiles, is better at building or storing soil carbon. And so we just got this data back on Friday. This is fungal isotope data. And so we haven't really had time to go through the data carefully, but what we're seeing is that when these families are mixed, so here we have 10 strains of Gigasperaceae and 10 strains of Glomeraceae that we've mixed. And we actually find first that there isn't a decrease in the proportion of fungal carbon that we find in the total carbon fraction. And so in fact, it's slightly increasing relative to the Gigasperaceae. And so we still need to do the colonization work to determine who is colonizing these roots. But assuming that Glomeraceae isn't 100% the dominant colonizer, what this might suggest then is that rather than a specific set of traits that might be exhibited by one of these families, that a combination or mix of these traits might be better suited for building fungal carbon that could go into the soil carbon fraction. So this brings me really to my final point, which is that we know that microbial communities exhibit this wide range of traits, and certain species have distinct characteristics or trait profiles, and that this varies across environmental conditions. And so in thinking about building soil carbon, should we be managing for a specific set of traits, or instead should we be trying to manage for an environment that improves the overall fitness or performance, and thus maintenance and continued biomass growth of the overall existing community? We know that the soil environment is incredibly heterogeneous, and that environmental conditions fluctuate over very small spatial and temporal scales. And it may be that you need all of those traits present in order to maintain fitness and biomass growth, and that it's not just one set of traits that we want to focus on for storing soil carbon. So with that, that's my summary, and I'll turn it over to Dr. Averill.
Thanks so much, Dr.
Sorry about the technical problems.
Well, it was great to see the data fresh from Friday. That's rapid turnaround. Thank you for that. And let's turn it over now to Dr. Averill.
Thank you, Bruce. Can you see my screen?
Yes, thank you.
Great. Okay. So my name is Colin Averill. My research team studies the forest microbiome. So how does incredible soil biodiversity affect which trees are in a forest, forest carbon sequestration, and climate change forecasts? We focus on the ecology of mycorrhizal fungi. So maybe there they are. There are friends. So these are fungi that form symbiosis with the roots of most trees on Earth. And these organisms make up what is sometimes called the wood wide web. Their living fungal hyphae can connect trees, allowing them to share resources, allowing trees to send messages and communicate. These fungi are also central to how nearly all trees access critically limiting soil resources. And I want to emphasize that there are thousands and thousands of species of these fungi, as Cynthia was just alluding to. And they have incredibly diverse strategies for how they access soil resources and how they engage with the soil environment. Yet until now, we really didn't have a good handle on how particularly which fungi are inhabiting a forest might affect carbon capture in tree stems and in soils. And so this is work led by Dr. Mark Anthony, a postdoc who works with me at ETH Zurich. And we wanted to know whether it mattered which mycorrhizal fungi live in a forest for the ability of that forest to capture carbon. And so we partnered with a continental forest monitoring network in Europe called ICP Forests. And we had foresters mail us soil so we could use DNA sequencing to characterize which fungi were living within the roots of each of these forest monitoring plots on this map. We then matched these taxonomic profiles of which fungi live there with fungal genome information, basically telling us which fungi are capable of doing which things in soil. And so we generated fungal trait profiles. And so we analyzed how mycorrhizal traits might affect tree growth and carbon capture. And so after controlling for climate and tree species and all these other environmental drivers, we find that the more a mycorrhizal community is dominated by species that invest more in inorganic nutrient acquisition, the faster that forest grew and sequestered carbon in tree stems. It was actually approximately 3 times faster. And we went actually to a handful of these sites. We sourced fungal inocula from them. And we were actually able to induce these effects in a common garden experiment in the lab, suggesting that these relationships may actually be causal, at least in seedlings. And in that, these seedling responses are really consistent with what we see in the field. And I want to emphasize that we could only really see this relationship by taking this really large scale approach. We really needed to observe at least 50 to 70 forests before these patterns could really emerge in a robust and a consistent way. And so our next step is to evaluate how these fungal strategies may be linked to soil carbon accumulation, taking advantage of repeated soil surveys that have happened over decades across this forest monitoring network. Yet these kinds of sort of long-term data on soil carbon accumulation generally just aren't available in most places. As a community, I think one of our biggest challenges is developing a way to better detect changes in soil carbon stocks over shorter amounts of time with less effort. If we could do that, I think we could rapidly discover management strategies and microbiome manipulations that can enhance carbon capture in soils. I think there is, some promising emerging technology out there like near infrared spectroscopy methods that could allow, for rapid characterization of soil carbon content in the field. And ultimately, the opportunity that this research suggests is that, soil biodiversity has a massive impact on ecosystem scale outcomes. And there's a potential capacity to manage or engineer this soil microbiome, the fungi and the bacteria and the animals and the viruses, to enhance carbon capture in ecosystems. I think we really shouldn't be so surprised that these organisms have such massive potential for ecosystem scale outcomes. Soils are the literal foundation of terrestrial ecosystems, and the microbial communities that inhabit soil are the most complex and most biodiverse communities on Earth. And for the first time, we're finally starting to understand which organisms live where? At really large regional, continental, and even global scales. And so imagine if we were trying to study photosynthesis, but over 90% of the time we didn't know, whether we were measuring a sequoia tree or a sphagnum moss. And then all of a sudden you did. And that's what's happening right now in global soil microbiology. And we should really expect this information and our emerging understanding of global soil biodiversity to really transform how we think about ecosystems in really foundational and transformative ways. And that's all I have. Thank you.
Wonderful. Thanks so much. What interesting content. Okay, can the panelists please turn on your cameras and microphones, and we'll enter the general discussion phase of our webinar today. There you are. Great. Okay, lots of good questions in the chat. And I want to start with one that's fairly general and important one to resolve. What is soil carbon sequestration is part one of the question. And Dr. Buckeridge, I'd like to hear from you on that, and everybody be thinking about the second part of the question, which is also interesting. What is the evidence that efforts to increase soil carbon sequestration are permanent enough to make a difference for climate change? So, Dr. Buckeridge.
Can you hear me? Yes. So soil carbon sequestration, I think of as net negative emissions. And so we're storing carbon in soils more than we're releasing it. And so it's a whole bunch of things. It's a change in practice, a reduction in emissions, and also potentially an increase in carbon, an increase in carbon storage through different mechanisms. And one of those is through, one of the easiest ones of the climate solutions is carbon sequestration, because it's something that in theory we can enact right away, as opposed to, for instance, carbon bioenergy, carbon and storage, or some of the other ones where we put biochar, things that need to have further testing before we can start using them on a regular basis. That's, somebody else might want to take over though and add more.
Yes, please. And that question about how do we know that it's going to last long enough to meaningfully influence climate change? Dr. Kallenbach, Dr. Averill?
I'll say something really briefly, which I think we have some evidence if we look into the past, but the real challenge now is looking into the future, where we have so much global change disturbance that's shifting the mechanisms that are storing carbon that in ways that are challenging to predict. So what happens, for example, as the Arctic starts to thaw, and what this means for that carbon and the plant communities and all those interactions is really challenging moving forward in terms of long-term stabilizations. I don't have an answer for that, but I think that's one of the bigger challenges with soil storage and evidence that it can be maintained into a really uncertain future.
I think the other important point is that, carbon does not need to be stabilized forever to matter for carbon removal and atmospheric carbon management. So when people working on carbon sequestration above ground, when we're looking at trees, there's companies that actually pay people to just defer timber harvests for some number of years, and just keeping that even for a couple of years longer in biosphere organic matter pools instead of in the atmosphere counts. And so that accounting is moving towards just, instead of tons carbon, it's tons carbon years, ton years. And so, temporary reservoirs are still reservoirs, and all of these things can contribute. And so we need to think about how much and how long, and those things go together. And if we have 10,000 carbons that we, keep down for 100 years, or, one carbon that we keep down for 10,000 years, those are actually kind of the same impact on the atmosphere. And so I would encourage you to, just because the carbon isn't permanent doesn't mean the sequestration doesn't have value.
And maybe can I also add to that no carbon is permanent anyway. It's just a relative scale, right? And I know you know that, everyone knows that, but it's just something to keep in mind, that because just because I brought up in the talk that I did about how mineral associated carbon, which is our kind of gold standard for stable soil carbon, it in itself is not permanent. Like I've done a study where I see it disappear in a year. And other times there's other studies that have shown it lasts for decades. So it really depends, actually. It is all relative.
That's right.
And
it's
there's
a cycle
uncertainty.
after all, right?
Pardon?
It's a cycle after all.
Exactly.
So another related question is that we haven't talked about methane, another important form of carbon that soils emit and consume. Is there, some thought about methane that should inform our discussion today? I'm thinking of earthworms too, Dr. Angst, and their propensity to shift the balance of gases that are produced from soils.
Yeah, actually, there's a meta study from 2013, I think, which states that earthworms actually increase N2O emissions from the soil and also CO2. There are also other studies where earthworms interact with other soil fauna to depress N2O emissions from the soil. So it's not even methane, I know, but regarding methane, it's like I think it also has been shown that earthworms increase methane emissions from the soil, but like the literature is relatively unclear, and it's also dependent on the study period, because most of the studies last like a maximum of 200 days, and what happens after this is like widely unknown, because scarcely any study investigates the effects longer than 200 days. So actually, in a rather moderate to short to moderate time frame, earthworms actually increase emissions from the soil, I think independent of CO2 or methane or N2O. Yeah. So,
and we heard also about rice today, and that's of course an ecosystem where methane production can be quite high. Any other thoughts on methane, or we can move on to the next question? Oh, this is a fun one for Dr. Buckeridge. Why is microbial necromass have lower energy than plant residues?
Maybe not than plant residues, than plant root exudates.
Oh, okay.
Because plant root exudates are pretty juicy for microbes. They're lots of easy sugars and amino acids, so they're less work for them to decompose, whereas the plant residues and microbial necromass are a little bit more work. They require the production of exoenzymes, multiple steps, sometimes different microbes working together in community to break it down. And yeah, that's why.
Yeah. Got it. Thank you. And a related question is, how might those exudates affect the stabilization or loss of microbial necromass? Now, a number of you touched on that idea in your presentation this morning. Dr. Kallenbach, would you be interested in taking that question, sort of the interaction between all those organic compounds in soil and the microbial necromass that's stabilizing or disappearing?
Yeah, so root exudates are tricky, right? Because we know that they can accelerate organic matter decomposition, and a paper just came out from Dr. Jillians out of Oklahoma State showing that root exudates can destabilize mineral-associated organic matter through both abiotic and biotic processes. But the other thing that root exudates do is they're soluble compounds, and not only are they rich in energy, but they're also presumably easier for microbes to access. And so we have sort of this tension between destabilization mechanisms and priming, but also a substrate supply for microbial communities that can build biomass and increase necromass production. And on top of that, then roots are also changing the chemical environment of the rhizosphere. And so we have all of these complex interactions, which is probably partly why the rhizosphere priming effect is so variable. because we have abiotic interactions, biotic interactions, and influencing biomass growth and turnover and recycling and desorption dynamics.
Yeah, I'm just thinking about how that rhizosphere priming effect is actually one of those things that I've lost sleep over for many years now, right? I mean, it's been such a mystery. So fascinating though. And I think we're making some progress. Okay, shifting gears a little bit. Focus on agricultural lands is important. Focus on natural lands is important. But the question is, there is a lot of land in cities, parkways, medians, parks, yards, gardens. How can this work be used to develop best practices in urban lands? What do you all think about that? The balance of it's accessible, it's right here, it's where we live, and compared to agricultural landscapes, maybe there's not as much. How do urban lands fit into this whole picture? Dr. Averill, would you be interested in taking that question?
Sure. So I think one of the movements that's happening right now that I think is really cool is just sort of this push towards, incorporating more aspects of native biodiversity into urban landscapes. So much of what we plant in cities is essentially, green furniture. It's stuff that isn't native to that place and can't really reproduce there, actually. It's not really an ecosystem. It's just stuff that you put in and rip out every year. But if you start growing things like native grasses, and I used to live in Austin, Texas, and that was a huge part of landscaping there, like thinking about things that are drought tolerant, but also build these huge root systems, or whatever you're doing, thinking about building an ecosystem that actually is native to that place and lives there is also going to reduce the amount of disturbance you introduce to the soil. And I think one of the things we know extremely well in soil biology and soil chemistry is that the less you touch the soil, the more carbon you're going to accumulate, the more organic matter that's in that soil, the more water holding capacity, the healthier the system you're going to have. And so I think, that sort of shift from treating our landscapes in urban areas like, furniture to treating them like ecosystems is the biggest shift we can make.
I like it. From furniture to ecosystems. That's really good. Okay, here's a question for all of you. I'm going to point to Dr. Angst first, though, for this. How do you measure dead biomass or necromass? And I'm thinking, Dr. Angst, in part about all your work with biomolecules and other things, that point to potentially ways you might measure this. What do you think?
Well, like a common way to measure microbial necromass is the extraction of amino sugars, which is like a cell wall component. This is present in dead cells or remains after the cell has died. This is a way you can also measure nitrogen compounds via NMR spectroscopy, but this is less targeted, I guess. Maybe Kate, Dr. Buckeridge can add more to this.
Yeah, please.
Sure. I mean, you're right. I mean, the things I put on the first slide that I had, that amino sugars was one. And then looking at different carbon compounds, and actually Dr. Kallenbach did a really nice paper looking at the different carbon compounds using, I believe it was GC pyrolysis. There's lots of different methods you can use to isolate certain carbon functional groups that are associated with necromass or carbon molecules. And then the best, my favorite, I think I mentioned, is growing up microbes on isotopically labeled substrate and then killing them and making isotopically labeled necromass and introducing it to the soil and tracing it through. You can follow it up with physical fractionation to look at that stable isotope necromass that's on the mineral associated soil as opposed to in the bulk soil to see where it was stabilized. Yeah, there's lots of fun things you can do. Then you can also measure the CO2 that's produced and see how that necromass breaks down into and is mineralized by microbes. So that's, maybe somebody else wants to add more. There's lots of ways of doing this.
There are lots and lots. I'll
actually add something to that because I'm not sure I agree. I don't think we have a good way to do this. It's very challenging to, in a natural ecosystem, isolate the entire contribution of microbial compounds to soil organic matter. We have proxies, but a lot of those proxies are shared with highly decomposed plant compounds. And We can come up with creative approaches in the lab, but translating them into a natural ecosystem, I think, remains one of our biggest challenges for this line of research.
It's true. And you think about how long we've struggled measuring biomass in soil, the living stuff, right? Necromass is even harder, but there are a lot of techniques that are at least pointing the way. And
I think we still struggle to measure biomass.
I know,
actually.
we do. Yes, yes, I agree completely.
Maybe
chloroform certification, right? Sorry, Dr. Angst.
Maybe something to add there. There's an approach how we can upscale amino sugar contents to the whole microbial necromass in the soil, which is indeed based on laboratory cultures. So it's questionable if it's really transferable to the field. But this is one way to somehow like have a raw estimate of the microbial necromass in the soil, apart from the proxies we have.
Yeah, yeah. Thanks. Great. Another question for you, Dr. Angst, about the earthworms. Are there, have we identified an optimal or suitable number or density or diversity of earthworms to derive their benefits in agricultural soils?
I'm not aware of any study who provided such an estimate. And it probably also depends on what you're focusing. So if you want to have a fertile soil, it's good to have earthworms because earthworms increase soil fertility. But as I said, it's like the effects on carbon sequestration are not that clear, also in the longer term. So I think it depends. So if you're a farmer and want to have fertile soil, then it's definitely not bad to have earthworms. And to add, maybe some ecosystems are also inhabitable by earthworms. For example, very acid forest soils, if you deploy earthworms there, they will die in a minute. So not in a minute, but like they will die.
Quickly, yeah.
Quickly, yeah.
Thanks. All right, a question first for Dr. Averill, and we'll see how it goes. So this is about the loss of soil biodiversity. Given the advance of monocultures inducing lower carbon sequestration, increasing erosion, what is the status of soil biodiversity globally?
So I think something I would love to know as a microbial ecologist is, what is the stat of biodiversity loss below ground in soils globally? I mean, I think we really don't know. Knowing, like, we have global surveys of the soil microbiome, but they're hundreds of samples, and that sounds like a lot, but it's really at a global scale, still not very much. Those things are getting a bit bigger now, but I think one of the scariest things going on is that we don't really know what we're losing. Like, there are some anecdotes though, and some reasons to believe that things are changing. So I focus on these mycorrhizal fungi, and they happen to form mushrooms too. And mycologists around the world for decades have been sounding the alarm of declines in sporocarp mushroom biodiversity. People have actually really quantified it in Europe, and it's, these surveys have lost half of their diversity. They think a lot of that has to do with nitrogen deposition, but there's just species that are no longer seen anymore. Yeah. So we're certainly losing it. And we also know in agriculture, industrial agriculture that we practice now, as we till soils, we annihilate these hyphal networks. We get a completely different community of mycorrhizal fungi living in a tilled versus no-till field. And so I think as we think about the importance of soil biodiversity and maybe even protecting it, I think actually there's a lot of opportunity in our agricultural landscapes that we don't necessarily have for, plant or animal biodiversity, but there is an opportunity for microbial biodiversity. And so thinking about, moving to no-till, we see increases in the biodiversity of mycorrhizal fungi, and there's also opportunity to begin to manage that community, to have more complex relationships. And, I guess the last thing I'd mention is that there's a huge market emerging right now of microbial inoculants. And they're focused on single species, silver bullet strains. And they're like, we're going to put this one thing everywhere. And it's sort of repeating the mistakes of above ground agriculture below ground, relying on really low diversity systems that are not very resilient, very susceptible to pathogens and extreme events. I think we need to really change that conversation to move towards biodiverse communities. I think we can still manage the microbiome to confer traits that we want, but we can also manage for local biota and biodiversity. So I think to directly answer your question, I think if we want to know the state of soil biodiversity loss, I think we need more measurement, but I think that's happening.
Yeah, that's right. Good. Okay, thank you for that. Here's a question about microbial, or the stability of microbial profiles over time. How long are those, say, community compositions stable? Does it stay roughly constant for like a decade? And what does that composition mean for carbon accumulation and loss? Who would like to take that one? Dr. Averill, go ahead, please. I saw you on mute, so.
So we've done some analysis on the temporal stability of at least fungal communities, because that's what I focus on. So that's usually the thing I know about. And the thing we see is there's remarkable within year, so seasonal turnover. You know, the relative abundance of different fungal groups changes a lot from spring to fall. And it's way bigger than the interannual turnover. So the turnover between years is actually pretty low. It seems that the fungal community living in like that forest over there, and you know, that 30 square meters is that fungal community year after year. And this is sort of consistent with what we know about the natural history of these fungi as well, right? Like people who forage mushrooms, like they have spots where they know this fungus grows there, this mushroom fruit's there. I don't tell anyone. And they like hand them down through generations, those spots, right? That implies that those fungi are actually, you know, they live in that place in the forest and they're there for even decades. I would suspect that the bacterial communities are far more dynamic. I think a colleague of mine, Zoe Werbin, has been using the NEON data where they actually measure 3 times a year every year to actually show that. That's an early result. So yeah, I think it depends on which organisms you're talking about, but some of them can be pretty permanent.
Mm-hmm. It's really cool to see that global scale analysis of diversity in soil advancing as it is. Here's a question for Dr. Angst. What impact does underlying geology have on soil organic carbon levels? For example, do clay-based soils contain more or less carbon than shallow, chalky soils?
Okay. Well, parent material determines soil mineralogy and how the soil's weather or what kind of minerals form during weathering, and consequently also how these minerals can stabilize carbon, for example, in organomineral associations. So, for example, soil with a high clay content has basically more reactive surfaces to stabilize soil organic matter than sandy soil, for example. So it has an influence on the capability of the soil to sequester carbon by stabilization on surfaces. And on the other hand, it partly also determines what grows in this soil, for example. So it determines parent material, also determines input patterns of plant materials, etc. So it's, I think you cannot say a clay-rich soil generally stores more carbon than a soil with less clay, for example. So it depends, I would say.
Yeah, and one of the interesting things that's come up this morning is that it depends on interactions with that biodiversity, those mineral interactions with living organisms in soil and their necromass seems to be an important area these days.
Of course, yep.
Dr. Kallenbach, would you agree with that?
Yeah, I mean, we see variation in carbon storage and destabilization even across different clay types and clays that and weathering as the degree of weathering of underlying soil geology. I guess I would also point out too, I think related to clay is pore size and pore structure and this idea of proximity to minerals and availability of substrates and the potential for interactions are also going to be influenced by the physical space of the system, which can also be a consequence of underlying geology. So yeah, big impact.
Thanks. Okay, question for Dr. Buckeridge. How important is microbial necromass for CO2 emission from agricultural fields in particular?
So I think that has a lot to do with the earlier question that I got about how much it's why it's a low energy substrate. So it depends how much necromass is there and how much the microbes are using it as a substrate, because that's generally how the CO2 is being produced when the microbes are mineralizing it. If I'm understanding the question correctly.
Yes, no.
And so partly, I don't know if I can tell you that. It's the carbon use efficiency, or the amount of how microbes recycle that necromass and use it to grow biomass versus release it as CO2 is huge. There's a huge variation across land uses. I did a study with a group that I worked in Lancaster in the UK, and we showed there was a huge variation in the carbon use efficiency of necromass recycling. So microbes eating other microbes, dead microbes. So I can't give you a straightforward answer. It varies by ecosystem type, and mostly it varied by climate. So the precipitation history was a huge control on that. Can I add something more to you about the seasonality question that Dr. Averill addressed? That I completely agree with what he said, but I just also want to give another perspective, having looked at seasonality quite a lot in both the tundra and in grasslands, that soil moisture was a really huge control on within and across your patterns. So whether that is through freezing or through drought, that led to big changes, and obviously in microbial activity we're aware of, but also in microbial community structure. And so future changes in soil moisture or changes in temperature patterns are going to have a big impact on how stable those communities are. So that's something I want to add to that.
Yes, thank you. No, important point. Okay, here's a question about biochar. Dr. Averill, would you be willing to take this? And anybody else, please chime in. The question is, we know that biochar is added to soil as an amendment to reduce sea loss, increase sea gain, and slow climate change. So the question, should I add biochar to my soil to increase soil carbon, and to what extent? It's sort of a very practical question about how we manage our lands, right?
Yeah. So I am not a biochar expert, and I will fully admit that. And to be honest, as a soil carbon scientist, I'm a bit confused by the literature, because there's great papers by David Wardle showing losses of carbon with the addition of biochar. It can go, not that it doesn't increase carbon in other scenarios. And the thing I guess I don't understand is, what is the predictive framework that tells me, what type of char will create a carbon gain or a carbon loss in which type of ecosystem? And I don't know, that maybe somebody else does who's smarter than me.
You're plenty smart, Dr. Averill. We can't study everything, right? We had a meta-analysis about the effect of biochar on plant growth that did show regional differentiation, a promotion of growth in the tropics, less so in the temperate zone. But I think you're right that it really depends on what kind of biochar you're talking about, what kind of system you're talking about. There are a lot of system dependencies. There are other thoughts, comments on biochar, and that it's a great question, because it gets to what to do, what people who are thinking about managing their own soils can do with a proposition that's out there these days. I don't see anybody unmuting. So, thank you, Dr. Kallenbach.
Yeah, I'm with Colin. I get a little confused too with biochar. And I think I also don't know very much about it, but I think of it as it's a way to sort of bypass the microbial component of soil carbon, in the sense that the goal is to lock away carbon in the soil through biochar. Now, there's some evidence to suggest that, it changes nutrient availability for the microbial community, and it could have indirect effects on the microbial community. But yeah, it worries me that it's kind of like treated as a potential storage bank for soil carbon, rather than thinking about the carbon cycle as a dynamic, transformative process.
Yeah. Right. Yeah, and I think biochar used to be thought of as simply stable. Microbes won't touch it. And we know that that's not the case. It's very, far more reactive than initially thought.
That's true. Yeah. Yeah.
Okay, here is a proposed grand takeaway. When we maximize plant growth and diversity, we're likely to maximize soil organic matter, stable organic matter in soils. True? False? Comments? We actually worked on this a little bit in the Cedar Creek system and found some evidence in favor. What do you all think?
True.
True. All right, we have a true. We could vote on this, right? Dr. Averill.
Yeah. I mean, it's so easy to waver, but why not?
Yeah.
Yeah, I mean, I think there's some truth to that takeaway. Like, you know, if you have a degraded system, like somewhere where like agriculture has been practiced intensively for decades, restoring that system and increasing biomass production is going to increase soil carbon for sure, as you increase inputs. But I mean, that's going to continue to a point. If you do a global analysis of productivity, soil carbon as a function of, you know, forest productivity or something, you generally won't find a consistent relationship. So it's not actually the least productive places like boreal forests are some of the biggest carbon stores in terms of soil carbon. But there is some evidence out there. You know, we've looked at tree diversity in temperate forests as soil carbon as a function of temperate tree diversity, and there's certainly a positive effect. So yeah, I think the input side of it is a bit more complicated than just more. But I think there's more and more evidence that the diversity side does increase soil carbon.
Yeah, and I feel like we're getting the tools to ask that question about microbial diversity and soil diversity as well. Dr. Kallenbach and Dr. Angst, did you have?
Yeah, maybe I can add my opinion. I would generally say it's true if we restore an ecosystem with the native community, native plant community, that carbon storage increases, but it doesn't necessarily increases the stability of this carbon. So it doesn't mean actually that the carbon in the soil is stable. So if there's a disturbance in the ecosystem, then the carbon that's additionally put into the soil is probably lost again. Because for example, in forest ecosystems, a lot of carbon is not stored in by the mineral phase or stabilized in a way, but is present as free particulate organic matter, for example. And if you cut the forest, then the free particulate organic matter inputs cease, and the rest is quickly decomposed, and carbon storage decreases. So I think we also have to look at the different carbon pools and where the carbon is in the soil.
Do you think if you changed or increased the diversity of a northern forest ecosystem, it would move more of that litter carbon or palm carbon into the mineral associated carbon fraction?
Well, did you ask me? Well.
So I guess that I'm spinning off of the chat comment. Does diversity increase soil carbon? Does it change how carbon is moved through the soil?
Yeah, it depends. I think if you have a coniferous forest and you put there deciduous trees, for example, with a higher lizard quality, then probably microbial necromass increases and is preferentially stabilized. So you could probably increase or at least alter the stability of carbon in the soil by increasing biodiversity. But I think it depends again. I don't know if this was a good answer.
Yeah, I'm actually also a little bit confused about it because like I think about it, how we talked about traits and properties that would increase necromass. Biodiversity isn't usually on the list as something that we think about that would increase necromass or soil organic carbon production, but I guess I'm asking the other panelists, is there a study out there that's looked at that? Because I don't know of one.
Well, that's what, I'm not sure exactly how to articulate this, but this idea that there's certain traits that are really good for building soil carbon. There's traits that are really good for allowing a community, a below ground community, in the same way it's true for plants, to maintain its fitness. So in a really stressful environment, you might have traits that are not very good for efficiency or growth rate or something like that. But it allows you to grow in that environment. So you might be inefficient, for example, but at least you're able to grow, whereas other members of that community might be dormant. And so I think that this focus on specific traits that are going to increase soil carbon is really, we need to think more about matching the traits with the environment and the potential for disturbance down the road, so that those that fitness can be maintained over time. And that's where I get a little confused with this idea of selecting for specific traits, because we know that, you know, especially at the species level, maybe not at the community level, but species have these traits because it confers an advantage for them in the appropriate environment. And so to say that those traits aren't good for biomass growth, it just doesn't add up always in my head.
There's a lot going on, that's for sure, right? But I think the general question about biodiversity in soil includes that element of traits, right? The traits are part of that diversity. So here's a slight shift of gears. It's another big question. What about viruses, phages, and their impacts on soil carbon cycling and soil carbon stabilization? I'm going to throw that out to all of you. Though, Dr. Kallenbach, you're unmuted, so you can go first if you'd like to. Viruses, what do you think?
I think I wish we had Dr. Firestone on, but so we spend like a day in class talking about viruses and carbon, and it seems to me that some of it depends on the life cycle of the virus, whether it's lysogenic or not, in terms of whether the cells are lysing. And it seems that the way viruses could impact carbon gets back to Dr. Buckeridge's research on, you know, rapidly potentially lysing these cells, so you get these sticky dead microbes at really fast timescales, speeding up the turnover of production of necromass. And then, of course, as we know, it seems like it can facilitate diversity through horizontal gene transfer. What that means for carbon storage, I don't know.
Other thoughts on viruses? We've seen a lot in the ocean in their role in carbon cycling there. And I think evidence is growing in soil as well. Dr. Averill.
Yeah, I was just going to say, I mean, I don't know the answer, but I think this research topic is incredibly cool. And yeah, I mean, we know, like Bruce just said, like these viral predation activities like are critical to understanding, you know, where pro and synchlorococcus in the ocean are in ocean MPP. And it wouldn't be surprising if this was going on in, you know, soil food webs as well. And it's just a harder thing to measure. You know, we don't have a primer for viruses because that's not how they work. But you can measure them. And then the other thing that it made me think of is there's a lot of cool work in, you know, fungal genomics looking at viruses. There's a lot of viruses in fungal genomes. And they often confer neutral or positive traits, not necessarily negative traits. It's really interesting. Tim James's group at University of Michigan works a lot on that. So yeah, I mean, I think it's this huge component of soil biodiversity that we understand like this much.
Yeah. Great. Let's step back a minute for this next question and think about what we're doing now. What can we do globally in terms of soil carbon sequestration, in terms of rates and amounts that will actually impact, you know, global atmospheric CO2 concentration? What can we do fast enough to, as one questioner puts it, to bend the warming curve in the less than 10 years that we have? Where do you think soil carbon sequestration efforts fit into all this? And Dr. Angst, I'd like to hear from you about this, because I know this is a particular, well, for all of the panelists, honestly, this is an area of major interest. You know, what are we doing to manage soil carbon for climate change?
Okay, so first of all, I think that carbon sequestration alone is not the solution to climate change. So there has to be done a lot more. And I think the first thing we can do is to stop ecosystem change, or just to stop degrading forests or cutting down forests, for example. So just to maintain ecosystems as they are, or as natural as possible, to maintain carbon stocks. Yeah, we can restore degraded lands by, like, I think primary or natural succession is relatively quick. So if we just leave the sites restore themselves, then carbon is also restored, which like reduces CO2 in the atmosphere. And regarding management, we could like do, for example, agricultural management, like not conventional, but no-till, for example, or like in general, less intense or invasive practices. That's probably already enough for the beginning.
Yeah, thank you. I'm curious what the other panelists think. I'm thinking of things like the 4 per mil initiative as well. Yes, Dr. Kallenbach. Oh. Either way.
Yeah, so I'll just bring in another perspective, which is this question of tillage. And I agree that the first thing we should be thinking about is halting the disturbance and degradation. Now we have a lot of soils that are already converted to agriculture that we'd like to see improved, but the tillage is a tricky one because there's also a lot of data to suggest that at depth that it's not always effective at sequestering carbon, where you get rather a redistribution of carbon instead of an overall total increase in carbon. So as you convert to something like reduced tillage or no-till, then you get higher carbon concentrations near the surface, but then carbon concentrations deeper in the soil are actually decrease. And so, and I think this varies by ecosystem. So we're still figuring out best management practices for the sole purpose of soil carbon, but I agree that I don't know that we want to design agricultural management systems just to sequester carbon. They have a lot of other services that we could tap into, and we need to think about this from a multifaceted, multiple ecosystem service perspective. And as Dr. Angst said, soil's ability to really put a damper on climate change through carbon sequestration is kicking the can down the road in some ways. So my favorite is cover crops. I love the idea of increasing diversity into a rotation and having living roots in the ground. It's the biggest, in my mind, one of the biggest issues with agricultural productions. You can institute as many best management practices as you want, but one of the biggest issues is once you go from a grassland or a forest into an agricultural system, your primary productivity decreases unless you're in California or Nevada and you're fertilizing and irrigating and pumping up primary productivity from what would otherwise be a desert situation in the summer. But point being is there's just less input going into an agricultural system. And so anything we can do to increase the time that plants are present in the soil, that's just an obvious strategy to me. I'll be quiet.
Maybe I could add a little bit to the confusion, because although I agree with what the order that you laid things out, Dr. Angst, that was really nice. And also what you were saying, Dr. Kallenbach, about the question we have about till, but I think there's also questions about cover crops, as much as I love them too, in concept. what about the nitrogen cost of cover crops? Like, we often don't think of nitrogen costs when we're thinking about carbon sequestration, and a lot of the practices that we use in agricultural land to improve carbon produces more N2O, and cover crops is not entirely guilty of that, but there are certainly some evidence that it could be. And so, I mean, it's really difficult. And so when you come to 4 per mil, I guess, like taking it back to the big picture, it's like an ideal scenario, but putting it in practice is something that I think is really challenging. So even I was saying like how this is one of our most immediate solutions to deal with carbon sequestration in agricultural land, but even our most immediate solutions are actually really challenging. So, you know, the hierarchy that Dr. Angst gave is a really good one, because save what we have first is really important. And let's work out the challenges of what we can do to sequester more carbon in the meantime.
Dr. Averill, I think you had a comment here as well.
Sure. Yeah. I mean, I think you all said it really well. I think Gerrit said it right off the top. Like, you know, we need to stop emitting first. Like, yes, we need to stop emitting, and then we need to remove carbon. But if we don't stop emitting, there's no amount that we can remove that's going to save us. But assuming we do that, yeah, it becomes really important. I think what you guys have said about cover crops and tillage is right on. I just, in our other managed agricultural landscapes, which is forestry, like, you know, we have really short rotations. They're like 25 years in southern pine. Extending those just a bit, like, is likely to increase carbon capture in those soils, in those organic layers that are building. And second, I would be surprised if there wasn't an opportunity for sort of microbiome management in these systems to build soil carbon. Like, these are the most biodiverse communities in the world. It would shock me if there wasn't, you know, a knob in there. But I think getting that, you know, really is really limited by our ability to detect changes in soil carbon accumulation rates. Like, I think everyone knows it's a remarkably difficult thing to do. And if we don't figure that out, we're going to be stuck on this for a while. And people are going to be doing things that increase above ground carbon sequestration with no idea how they're affecting below ground carbon sequestration. And we're going to only value that above ground part. And we won't know if we're actually creating additivity or not. And so I would love to talk about that. Like, how do we stop, you know, grinding up soil and wrapping it in tin capsules and putting it in the elemental analyzer? And how do we get those numbers and those rates faster?
You're taking me back to lots of time with those elemental analyzers and those little tin capsules to measure soil carbon. Okay, here's another question with some historical perspective. No one has mentioned the role of soil biodiversity in the production of humic substances. So the question directed to Dr. Angst, for starters, has there been evidence for the production of humic precursor molecules by earthworms?
Well, some say that earthworms add like humic substances to the soil, which others call mucus. I would be like cautious using the term humic substances, because they're operationally defined in a mixture of all, more or less, of all compounds of organic matter. So I think it's questionable if it's a relevant or like a useful concept to further soil organic matter research. But yes, earthworms add mucus to the soil. That's true. In this way, they stimulate the microbial community. So this is actually true. Was this the answer to the question? What was the question again?
Yeah, I think so. And it's getting at kind of the shifting paradigm in our understanding of soil organic matter, right? Humic substances, the operational definition. And then there's another question that touches on this as well, arguing we spent decades understanding how growth, physiology, and chemistry influenced organic matter, only to realize that it's multifaceted, depends on many factors, not just the plant inputs, but also microbes. So now, on reflection, do we need to be careful about the potential for tunnel vision about microbial necromass as the answer? You know, which I admit I was guilty of in my introductory comments. I think partly it's really exciting to think about it that way. It is a new perspective. But the question is, you know, are we likely to repeat mistakes of the past in terms of our tunnel vision on having the answer with this focus on microbial necromass? What do you think? Dr. Buckeridge, do you have a a thought on that? Are we tunnel visioned?
I think we're just excited. It's kind of like, you know, when you go to Minesweeper and you hit like the thing that it opens up the big new area. It's sort of like that. It's like all these new questions and things that we can ask. And maybe one of them is that secret bullet to have greater understanding, because we've done so much, as the person who asked the question said, on plants and plant decomposition. But I don't know if you can remember the figure that I had in my talk. Plants are still really important, and plant material is still a really, like, not only does it feed the microbes, but it's still also in itself preserved. It's not only necromass that's preserved on mineral surfaces. There's still plant material that's preserved there. So it's still important. It's just that it's changed our understanding, and so we're excited about it. Maybe there's a tunnel vision. There's certainly been a lot of necromass papers out in the past year. They're exponential right now, but that's okay. I think like we need those. We need a greater understanding, and yeah, I can't predict what will happen about our tunnel vision, but I think it's just making our understanding stronger. So it's a good thing.
I agree. Other thoughts on that?
Yeah, well, I also agree that the necromass is like more important than thought before, microbial necromass to solid organic matter. But we've done a review recently, and a lot of ecosystems are still like not necessarily dominated, but like very much influenced by plant compounds, which are also stabilized in soil. So it's not just microbial necromass; it's also like still part of the old paradigm that there are also plant compounds like lignin, lipids, whatever. So I guess we have to focus not only on necromass, but on everything. So, but probably most people have this in mind. I don't know.
Yeah, that's right. We're coming close to our end time, and I just want to throw out one last question. I thought the presentations today were really fantastic, and there's a question that's pushing us further on that. How do you see this type of information being simplified and communicated to landscapers, gardeners, commercial growers, and the like to get them on board? What do you think we can do more of in this area of communicating this complexity? We should have counted how many times we as the scientists said, oh, I'm confused about that. I mean, a lot of this is cutting edge, and yet there are some clear take-home messages. So what do you all think we can do to do more? Dr. Kallenbach, I'm gonna pick on you first for this one. What do you think we can do more to simplify the communication about the importance of biodiversity and soil for carbon?
Well, I think that sometimes we do a good job simplifying the message. Maybe not today, but in certain venues, for example, on farmer panels, for example, or in class, or talking to high school students, I think that there has been effective messaging in terms of just improving the overall health and diversity of putting the focus on the microbial community in terms of its importance of interacting with plants and the manifesting the soil services that we want to see, whether it's carbon storage or plant fitness, plant health. So I'm not sure that I'm not sure that we are failing in that area. And I sometimes talk with farmers, and it's impressive what they take away. They're just like, I need to feed the microbial community. Okay. And we know how to do that. We know how to do that, and what types of management is not good for that. So I think we're on the right track, and I'm quite optimistic.
Thank you. And to clarify, I thought the presentations today were really brilliant, very clear, interesting, provocative. It's when we get into the questions and the details and dig in literally to the ideas that we end up, I'm not sure about that. There's always more work to do, but thank you. Thank you all for participating today. And there are many questions in the discussion that we missed, but I see we are over time, I believe now. Is that right? Is it time to turn it back over to Juliet? Thank you. Thank you all for tuning in today and listening to the conversation. And again, the event's been recorded. It will be available on YouTube soon, and wishing you all the best in your investigations and curiosity about soil biodiversity and carbon. Thanks.